Okay, this gives me a chance to mess you up for your future endeavors. A tremendous amount of biologists are very limited in their understanding of the world because the majority of biologists are very vertebrate-centered, very animal-centered. And so they don't quite, they don't have the breadth of experience to understand that some of the stuff that they were taught many, many years ago doesn't really apply to everything. Like, I can't tell you, you can look at, if you put definition of asexual reproduction into Google, most of the time you will get a definition that doesn't fit the definition that I gave you, because it's too much based on a very simplified, gendered version of sexual reproduction, and that asexual reproduction is just not that. So it's reproduction without two parents. Well, so is sexual reproduction typically doesn't have two parents. Or they'll define sexual reproduction as involving sperm and egg, which it doesn't in funguses, it doesn't in many, many, many organisms. But they didn't learn it that way, and a lot of times they have never even run into those other exceptions to realize that their definition that they're giving as a basic one-size-fits-all definition doesn't really fit all. Telophase and cytokinesis, they've got them lumped together here. So whoever put this together went, oh, that's when it happens. Well, maybe they're just trying to show the two together as you know, because that's the logical place to put it, but it is misleading. So now we're going to pick up the rest of mitosis. Once you get the chromosomes in the middle, it's metaphase. That looks like a line, doesn't it? That looks like a line, doesn't it? But it's in a three-dimensional cell. So when you see this in the right orientation, it's really the only orientation where you can see it, then it is metaphase, but it isn't really a two-dimensional line. It's a three-dimensional plate. And in fact, one of the first biology books I taught from had a circle of chromosomes labeled as prophase, but you couldn't see the spindle. So once these things are in the middle and they separate out, these are also three-dimensional objects in a three-dimensional cell. If you can see a circle of chromosomes, but you can't tell where the spindles are, you could be looking at prophase. You also could be looking at anaphase turned 90 degrees, or you could have cut through one of these groups or metaphase or anaphase. Without seeing the spindle, you don't know which it is. So the world gets confusing. And even, you know, picking a picture to put in a biology book, somebody didn't think that through. So the spindles are, not all of them connected to the chromosomes, because there's a lot of spindles and not that many chromosomes. You got to throw a whole bunch of them out there to just get any of them. And once they're in the middle, you are set up. All we need to do is get the copies away from one another. Because it turns out once you pop that centromere apart, this side of the centromere is connected to that set of spindles, and this side is connected to that set of spindles. And once there's no pull in the opposite direction, they'll start to disassemble at the centrosome end and get shorter and shorter and shorter, and they move the chromosomes away from one another. Now, some cells get into metaphase and stall there for a while. You would think metaphase would go by very quickly, and it usually does, right? You just get them lined up, they're in metaphase, pop them apart, they're in anaphase, but different cells do this on different paces. So you can't always say, well, metaphase got to be the shortest stage because of what happens during it. Because any stage for some cells, they might be, that's a good place to wait for a while. So now you have single-strand chromosomes, no longer chromatids because they're disconnected, being hauled by their centromeres toward this microtubule organizing center, the centrosome. As soon as there's a gap, you're in anaphase. And as soon as the cell is ready to do it, it will start to reconstitute a nucleus around those chromosomes. Now in plant cells, they actually start to build a cell wall across the middle of the cell, because remember, plant cells have walls around them. And the two new cells are going to need walls. So typically, and this isn't true for all plant cells, but typically, they start to form bits and pieces of a new cell wall across the middle, and that's called the cell plate, because it isn't complete yet. So it's bits and pieces, gets a different name. Some animal cells will start to split as soon as the chromosomes get separated from one another. They will start to go through cytokinesis in anaphase. In fact, for our animal cells, for the mitosis lab, what we call telophase isn't really telophase, but it's the only late stage that you can recognize through a microscope, because in the fish cells we look at, they basically are already divided before they really reconstitute back into interphase. That's where this is headed, right? Back to an interphase nucleus, or two interphase nuclei. So all of the stuff, this is telophase. Metaphase lined up in the middle. Anaphase separated out, and various things. Telophase, let's put stuff back the way it was. So it's essentially metaphase backwards, but it may not be backwards step by step by step by step. Yes, you need the nucleus back, so the nucleus reforms. Yes, you need a nucleolus back, it reforms. Yes, you need the chromosomes spun out so that you can use them, so that happens. But whereas the nuclear membrane may have disappeared early in prophase, it may appear early rather than late, if you're going opposite, in telophase. So it doesn't necessarily reflect. Step one in prophase isn't necessarily the last step in telophase, and working their way back that way. But everything kind of goes back to the way it was. In prokaryotes, remember, prokaryotes have a single loop chromosome. It's supposed to show the differentiation between plants and animals. Here's the cell plate. Not a huge amount of difference. And it's only true for the types of stuff that we use. So prokaryotes, we'll slip prokaryotes onto this video. Prokaryotes have a single loop chromosome. They make a copy of it, and the two copies separate. They come separate from one another. Now, somebody probably knows more about this than I do. I've been doing some background research trying to find this stuff out, and I haven't been able to locate information about this. But, so this is now a bacterial cell. Over here on the inside of the membrane, and over here on the inside of the membrane, are two little hooks that will hook onto the chromosome. So far as I can find out, they don't have a set name for those, which is weird because people love to name stuff. But if there's a name for these two little hooks, I haven't been able to find it. But the new loop chromosomes need to be attached at both sides, because that's their message that separation of the duplicates has occurred. And once you've got a loop chromosome over here and a loop chromosome over here, you can divide down the middle of the cell into two bacterial cells, and each one gets a complete chromosome. Now remember, bacterial cells also have plasmids, little mini chromosomes with a lot of times genes that are on the main chromosome. Sometimes it's a gene you use a lot, so you make extra copies so that you can crank out a lot of protein. Sometimes it's a plasmid you picked up from another cell, and it may have an allele that you don't have that's very useful, like antibiotic resistance, or sometimes a whole gene that does something that you weren't able to do before. And remember, that's the kind of quasi-sexual reproduction that bacteria are capable of where they can produce offspring that are genetically different because of the way they swap around genetic variations with one another. But it's a different, they don't go through the same stages. Well, for one thing, they're not making a new nucleus, but they do need to make a copy of the chromosome. If they're gonna make a new cell, they're gonna make sure that a copy goes into each of the two new cells, and they have a very simple way of getting that done. And that's, let's see. Yeah, I'm gonna do meiosis on another video.